quiet night until a strange letter arrived. No fingerprints, no suspects, just ink.
Chromatography is a technique used in laboratories to separate components in a mixture. It involves two phases, a mobile phase, where the mixture is dissolved in a fluid, and a stationary phase, which is a solid material the fluid passes through. Different substances in the mixture move at different speeds depending on how strongly they interact with the stationary phase. Those that bind more strongly move slower, leading to separation based on their physical or chemical properties. Common types of chromatography include thin layer chromatography (TLC), paper chromatography, high performance liquid chromatography (HPLC), and gas chromatography (GC).
In forensic science, scientists analyze many different materials like ink, lipsticks, and even explosives to help solve crimes. Because every material has unique chemical properties, different methods are used to identify them. One common method is planar chromatography, especially thin layer chromatography (TLC). This method is used to check things like inks, example on fake banknotes and forgeries, dyes, and drugs. It works by letting the sample move on a thin layer and each substance travels at a different speed. This helps to tell them apart. TLC is quick and cheap, but for more complicated cases, other advanced chromatography techniques are used.
The only clue left at the crime scene is this handwritten note. Let's find out which pen was used.
Chromatography helps forensic scientists analyze ink ransom notes, drugs in bloods, explosives and poisons, and dyes in fabric or hair.
Let's break it down. In forensics, chromatography starts by collecting a sample from the crime scene. The sample is then prepared by dissolving it in a suitable solvent. After that, the sample is placed on a surface like paper, a plate, or inside a column. A liquid or gas called the mobile phase is added to move the sample. As it moves, the different parts of the sample separate. The results are made visible using light, stains, or special detectors. These results are compared with known substances. Finally, the findings are recorded, analyzed, and reported for use in court. The result is a chromatogram, which is a color pattern unique to each ink.
The video shows the chromatography process we conducted to identify the type of pen used in the letter. First, we prepared a strip of chromatography paper and applied small dot of ink from several pens, including the one from the letter. Then, we placed the paper into a beaker containing sodium chloride solution, which served as the solvent. As the solvent moves upwards through the paper, it carries the ink pigment with it. Because different ink are made from different combination of pigment, the separation form unique patterns called chromatograms. This technique is especially useful when dealing with forged document, anonymous letter, or any case where ink analysis is required. It allows investigators to identify which ink or pen was used, even if the writing appears similar. We are comparing the chromatogram found in the letter with several ink samples. After analyzing the pattern, we discovered that the ink from the letter perfectly matches the chromatogram of pen B. This proved that the suspect used pen B to write the letter.
Match found! We just cracked the case with chemistry.
For our information, rare cases where chromatography helped are detecting drug use in athletes, identifying forged signature, and analyzing explosive residues.
It's not just TV. Chromatography really helps solve real crimes.
In forensic science, every detail matters. Chromatography helps make those details visible. Why chromatography? It is because high accuracy, low cost, and works on complex mixtures.
In the end, chemistry doesn't lie. Case closed.
It's the motherfucking Eagle Double G. Snoop Dogg.
Chromatography is fast, reliable, and precise.
In forensic science, the smallest chemical detail can reveal the truth.
From crime scenes to labs, chromatography uncovers the truth, one molecule at a time.